So this is the nervous and sensory systems. One of the organ systems of your animals. We talk about the nervous systems and the sensory systems ahead. For communication to happen, okay, the animal has to integrate body functions and homeostasis, which are released by two factors. So first one is the neurons. Okay, neurons are transmitting electrical signals, and they give information to create a quick response to a certain tissue or a certain cell. And hormones are slower or chemical signals that initiate a widespread, a prolonged response in a variety of tissues. So we will be talking about the functions of your neuron, the anatomical organization, and the evolution of a nervous system in animals, and the ways that senses and collect information and transmit it along the nerves of your central nervous system. So what are the neurons? The basic functional unit of your nervous system. So this is called a neuron because they produce signals, and it is communicated over short, relatively long distance. And what are the functions of these neurons? It has two important properties. The first one is excitability and the ability to respond to stimuli, and the second one is conductivity, the ability to conduct a signal. So we have functional types of neurons. So you have three functional types of neuron: the sensory neuron, interneuron, and the motor neuron. So for sensory receptors or afferent neurons, they act as receptors of stimuli themselves and acting as receptors. Okay, they will be activated by receptors. So if there are changes in the internal or external changes or that stimulate sensory neurons, they are sending signals to the brain where the information is being processed. So these are basically coming from your senses. Okay, your vision, olfactory, gustatory, tactile, and auditory. These are your sensory neurons. And the interneurons are the ones who receive the signal and from the sensory neurons and transmit them to motor neurons, which are the effector or efferent neurons. So they will be sending the process information via a signal to the body effectors. So example for that is your muscles, causing them to contract, or to glands, causing them to secrete glands. So we have the figure 24.2, which will summary the flow of the information in your nervous system. So these are vertebrate neurons. The brain and spinal cord will create the central nervous system of the vertebrates, and sensory and motor neurons from the peripheral nervous system. So we have touch, taste, smooth muscles, skeletal muscles, and within the CNS, the interneurons, okay, this one here, they will provide the link between the sensory and the motor neuron. So these are the touch or taste. So going to the motor neurons in this part, they will command via using the impulse to your muscles or glands or the effectors of the vertebrate. So this is a pathway of the information of the nervous system. We have the input stimulus, and it will be going to the receptor, the sensory neuron, and then the intermediate between the two, the interneuron, will send the information from the central nervous system going to the motor neuron, to your effector, the muscle or your gland, for example, and the output response. So what are the key functions of your neuron structure? Your cell body, okay, of the neuron has a large central nucleus. So going to the next figure, it has many short thread-like branches. We call them dendrites, and they are actually extensions of the cell body, and they are named after the dendrites or dendron, which means the tree, because they resemble like a tree. Okay. The axon is also a long cylindrical process and conducts signals away from the cell body. So for the neuron, okay, the language or the signals of your neuron is carried out by nerve impulse or your action potential. So if there is a key nerve impulse to your neuron's plasma membrane in its properties, there is a change, okay, in membrane permeability and subsequent movement of ions, which will produce a nerve impulse. They will travel along the plasma membrane to of the dendrites, cell body, and axon of each of the neuron. So we have this what we call as the resting membrane potential. Okay, um, the resting neuron is not a conducting nerve impulse, so they rest. Okay, so when the resting neuron is resting, it has a polarized plasma membrane, and the fluid on the inner side of your membrane is negatively charged with respect to the outside fluid, which is positively charged. You will see that one in the figure 24.3 here. In a voltmeter, will measure the difference between the electrical potentials between these two electrodes. So when an electrode is placed inside the neuron at rest, one is placed on the outside. So the electrical potential of this letter A is negative 70 MV, okay, which is relative to the outside. And in the neuron at rest, the sodium is more concentrated. Outside the cell, look at this one, sodium. And potassium is more concentrated inside the cell. A neuron is in the resting position and it is said to be polarized. Going back, okay, there is what we call as resting membrane potential, which is the difference in charges. Okay, and all cells have resting potentials, but neurons and muscle cells are specialized to transmit and recycle it rapidly because they're working with the nervous system, central nervous system. So we have these ion channels and the sodium-potassium pump. So we have to maintain the balance, the sodium and potassium from the both sides of the membrane, and thus it creates a membrane potential. So some of them are open, but others are open or closed by the position of your gates. So whether a gate opens or closes, a channel depends on the membrane potential. Such gates are voltage regulated. Some of these membranes are specific for sodium ions alone, and others are specific for potassium ions. So graded potentials these are the transient or changes in the membrane potential from a resting level to produce an electrical signal. So these signals are occurring in two forms. So graded potentials are important in signaling short, distances, whereas action potentials are for the long distances. Remember, graded potentials for short distance and action potentials for long distance signals of a nerve or muscle membranes. So what are graded potentials? So aside from the first definition, they are confined or changes in the membrane potential in a relatively small region of your plasma membrane. So they are called graded potentials because the magnitude of your potential can vary or is graded. Okay, they're usually produced by a specific change of the cell's environment acting on a specialized region of your membrane. So we'll talk about the invertebrate nervous system. So invertebrate nervous systems are the precursors of the nervous systems among the animal kingdom, and we'll take a look at the most basic nervous systems. Okay, so nervous systems of your invertebrates, the development of nerve net is highlighted, okay, as in Hydra. So the second trend involves cephalization, which is concentration of all the sensory organs and the brain, in the brain case, on the anterior portion of your body. So the nervous tissue and receptors in the anterior of the animal, like the flatworms, has exhibited such cephalization. So the third trend is a bilateral symmetry. So we have the equal portions of your left and right parts, okay? And the development of your ganglia represents the fourth trend. The fifth trend is the development of the invertebrate nervous system in an increased number of the interneurons found in your brain. Let's say for the vertebrate brains. So for number one, letter A, it's Hydra. It has nerve nets. Try to look at the sensory cells in your tentacle. Letter B is a planarian flatworm. It has an eye spot, and it is bilaterally symmetrical. And C is an earthworm, an annelid worm. It has a nerve in the brain and ventral nerve cords with ganglia. A crustacean, a crab, which has a principal ganglia and a visceral connective tissues. And letter E is a mollusk, a squid, which have brains and behavior complex than compared as those of fishes. Okay. Letter F, a cross section of your C star arm. Okay, so these nerves of ectoneural system terminate this on the surface of the hyponeural system. These two have no contact. Okay. So for vertebrate nervous systems, it's very obvious that bilateral symmetry, a notochord, and a tubular nerve cord characterize a evolution of your vertebrate nervous system. This is a cross-section in the gill region of your amphioxus, a lancelet coming from phylum Chordata, okay, in 40x magnification. Notochord is a mesodermally derived tissue, okay, a film sheet that lies ventral to the neural tube. It's found in here of the animal, the belly part. Okay, it first appeared in marine chordates and present in all vertebrate embryos, but it's greatly reduced and absent in your adults. So they are during the embryological development of your most vertebrate species. These are arranged, okay, in a vertebral column or placed the notochord. So for mammals or for humans, you'll see the spinal cord being housed inside the vertebral column. This is a larval stage of a tunicate in Urochordate. Okay. So these are your figure for the basic organization of your nervous system in all vertebrates. Okay. So we have the nervous system, the CNS below, and the PNS above. Under the CNS, we have the spinal cord and the brain. Under the PNS, we have the motor or the efferent nerves. We have somatic nervous system or the voluntary, and the autonomic nervous system or the involuntary. The sympathetic, the parasympathetic, and the enteric division are on the ANS. We have visceral sensory nerves and somatic sensory nerves that are part or give rise to your sensory afferent nerves, and that makes up the peripheral nervous system. Okay, so the spinal cord is linking the brain, okay, and most of the body getting involved with this spinal reflex actions. So this reflex is predictable and involuntary response to a stimulus. So both the voluntary and the involuntary movements of your limbs depend on this link. And the spinal cord is a part of the central nervous system, which extends from the brain near or to the tail. So we have it has been enclosed with the vertebra for the axial skeleton of your of a mammal. Okay, starting from the cervix, cervical vertebra, and then going into the sacrum or sacral vertebra. So we have the spinal cords of the vertebrates here. The first one is from a lamprey. It's flattened. Take a look at it. And it has no myelinated axons. Its shape will facilitate the division of gases, nutrients, and other products. Okay. For B and C, the shark and the amphibian, the spinal cord is quite longer. Okay, you look at this vascularized matter here and rounded here. And it has more of the white matter, and the spinal cord is bulging outward. So it has also the gray matter of your spinal cord. Letter D is a reptile, and E is a bird, which has a characteristic of a butterfly shape. So we have spinal nerves, okay? They are directly related to the number of segments in the trunk and tail of your vertebra. For example, a jump has evolved its hind legs for jumping and swimming, and a reduced trunk and no tail in your adult. So for snakes or reptiles, it has lateral undulations of its long trunk and tail, which has several hundred pairs of spinal nerves. So there's a lot of it found in your snakes, so for instance, the reptiles. So brain, okay, located on the anterior part of your spinal cord, undergoes a regional expansion of a hollow tube of nervous tissue and develops the hindbrain, the midbrain, and the forebrain. So the central canal of the spinal cord is extending up into the brain and expands into chambers. We call them ventricles, and they are filled with cerebrospinal fluid. So we have the divisions: the forebrain, the telencephalon and the encephalon, the midbrain, the mesencephalon and the hindbrain, the rhombencephalon. So these are the major subdivisions of the and the structures they contain. So this figure is flattened and simplified, and these are the main components. Take a look at it. So there's a lot of components found in the forebrain, then the midbrain and the hindbrain. So let's talk about the hindbrain. So the rhombencephalon, okay, it's continuous with the spinal cord, and it includes the medulla oblongata, cerebellum, and the pons. Okay, the medulla oblongata is the enlargement of your spinal cord, which will enter the brain. Okay, what are the purposes of these reflex centers? For breathing, swallowing, gastrovascular function, and gastric secretion. So medulla oblongata is well developed in all jawed vertebrates, including mammals, reptiles. Okay, and we are the ability to control its visceral functions serves as a screen of information that leaves or enters the brain. Cerebellum, okay, is the outgrowth of your medulla oblongata. So this coordinates motor activity with association with limb movement, maintaining your posture or spatial orientation. So the cerebellum of the cartilaginous fishes has distinct anterior and posterior lobes. So in teleos, the cerebellum is large in active swimmers and small in relatively inactive fishes. So for amphibians, we have a rudimentary cerebellum reflecting their relatively simple locomotor patterns. So these are the several vertebrate brains. Okay, they are not drawn to the same scale, but the evolution of the vertebrates with complex nervous systems resulted in the enlargement of cerebellum and the folding of your cerebral cortex. So take a look at the size of the cerebellum in the frog, so small, compared to the alligator, chicken, and for the cat. So it developed and grew, it experiences growth as you go ahead with mammals. Midbrain is the originally center for the reflex and responses to visual input. It also added functions as it evolved. So we have tactile, auditory, and it does not change in size. So the roof of the midbrain is called the optic tectum, which is a thickened region of gray matter and integrates visual and auditory signals. So for the forebrain, okay, it has two main parts, the encephalon and the encephalon. So for earlier fishes, they have only two lobes of brain's evolution started from a forebrain from fishes, and its main purpose is for olfactory. So the diencephalon lies just in the front of the midbrain, and it contains the pineal gland, the pituitary gland, the hypothalamus, and thalamus. So the thalamus will relay all sensory information to higher brain centers. And hypothalamus is also a part of the endocrine system. It is located below the thalamus and regulate many functions such as temperature, sexual drive, carbohydrate metabolism, hunger, and thirst. And also pineal gland controls some body rhythms. Cranial nerves, okay? In addition to the paired spinal nerves, the peripheral nervous system of vertebrates have paired cranial nerves. For mammals, birds, and reptiles, we have 12. Okay, for fishes and amphibians, we have 10 first pairs. For optic nerves, which are containing only sensory actions, they carry signals to your brain. So other contain sensory and motor axons, which are termed mixed nerves. Okay. So here are the cerebrum in different vertebrate species. They increase in size and complexity as your neural connections in more advanced group, like for humans, chimps, macaque. Okay. For fishes and amphibians, they lack cerebral cortex. Whereas the reptiles, the pit viper, and birds have a small amount of gray matter covering their cerebrums, and the cortex has covering their cerebrums. The primitive mammals like the opossum and carnivores such as the bobcat have large cerebrums, and the cortex has a few convolutions. For the primates, we have the cerebrum much increased relatively to the other brain structures, and the cortex is highly convoluted. And lastly, the humans, it dominates the brain evolution, which is highly convoluted brain. So we have autonomic nervous system. So we have two divisions that act antagonistically, right, or in opposition to one another to control the body's involuntary muscles, so smooth or cardiac and glands. We have the parasympathetic division and the sympathetic division. So let's talk about the parasympathetic. So it functions during relaxation. Okay, it contains nerves that arise from the brain and sacral region of your spinal cord, and it consisting of the network of long efferent nerve fibers that synapse at ganglia in the immediate vicinity of the organs, and short efferent neurons that extend from the ganglia to the organs. That's the parasympathetic. The sympathetic is for the flight or fight response. Okay, it contains the nerves that arise from the thoracic region or thorax and lumbar region of the spinal cord that's found in here behind your ribs. It's a network of a short efferent central nervous system and fibers that extend to ganglia near the spine, and long efferent neurons extending from the ganglia directly to each organ. So this is your simple diagram in your sympathetic division on the left, and on the parasympathetic region on the right. So we have the thoracic and lumbar areas of the spinal cord. So the ganglia are located near the spinal cord. The ganglion chain here. Okay. So it exits the brain here, the sacral region of your spinal cord in the parasympathetic, and the ganglia are located near the organs they innervate. So most of the internal organs are innervated by both divisions. So if the sympathetic will dilate, the parasympathetic will constrict. If the parasympathetic will secrete saliva, there's a negative opposing is to stop secretion. Constrict, dilate bronchioles, slow down heartbeat, speed up heartbeat, and vice versa. Okay. So what are sensory receptions? So sensory receptors are cells that can convert the environmental information into a nerve impulse. So for a stimulus is an energy or form of energy that an animal can detect using its receptors, and all of the receptors are transducers. They convert one form of energy into another. Okay, and all nerve impulses are the same. They are different to types of receptors and convert different kinds of stimuli, such as light or heat, into a local electrical graded potentials. We call them graded potentials. So if this graded potential reaches the sensory neuron's threshold potential, it causes the changes in your plasma membrane. So what are your sensory receptor features? First is they contain sensitive receptor cells or finely branched peripheral endings of sensory neurons that respond to a stimulus, creating a graded potential, and their structure is specific for certain kind of stimulus. The receptor cells synapse with afferent nerve fibers that travel to the central nervous system along specific neural pathways. And the CNS, the nerve impulse is translated into a recognizable sensation such as a sound. So we'll talk about the invertebrate sensory receptors. So your animal's behavior is largely a function of the responses to environmental information. So for the basic animals, the receptor structures through which they receive information about the environment and their coordination, okay, will be under in this perspective. So first, for animals, invertebrates that are sense changes in pressure, we call them baroreceptors. Okay. Examples for that are the ocean-dwelling animals. We have the copepod crustaceans, ctenophores, jellyfish, and squids. Okay, so they respond to the water depth changes and using their baroreceptors or pressure changers. In response to your chemicals, we have chemoreceptors. Okay, and the oldest universal sense in the animal kingdom. Some protozoa has it. Okay, they avoid basic or alkali salt stimulus and specific chemicals that attract the predatory ciliates to prey. Some aquatic invertebrates are located in pits or depressions through which the water carrying the specific chemicals may be circulated. And chemoreceptors are found in your antenna, mouth parts, and legs to form hollow hairs containing chemosensory neurons or cilia. This is an example of an invertebrate chemoreceptor in its cuticle, a dendrite endings, and it has a wall of hair and pore at the tip of your hair. Georeceptors, from the root word geo, which means earth, and receptor, it responds to the force of gravity. So up to down, so that's an animal's orientation of the pulling of the gravity. So the most georeceptors are called statocysts. Okay, so they are found in these are cilia bearing sensory epithelium within the chamber of a solid granule called the statolith. So most of them are found in your ear. Okay, a statocyst cross-section consists of a fluid-filled chamber and a solid granule called statolith. And there are some sensory epithelium containing non-motile cilia which detects the movement of the statolith and maintaining the balance or the georeceptors. For invertebrate phonoreceptors, these are using the voice, okay, or the phone. And it is demonstrated highly in insects, arachnids, centipedes, and other invertebrates seem to respond sound-induced vibrations of the substratum. So this includes crickets, grasshoppers, and cicadas, which possess phonoreceptors called the tympanic or tympanal organs. So it is like a drumhead principle. Okay, the flatter wall or tympanum of each trachea functions as a drumhead. As the tympanum vibrates in response to your sound waves, kasi phonoreceptors, the pressure will change. Okay, in the trachea that affect the sensory neuron and causing a generator potential. So the leg of your tibia of a cricket lead to the tympanal cavities. So photoreceptors are next. These are animals that are sensitive to light. So they have light sensitive pigments, we call them carotenoids and rhodopsin. So these are absorbing photons from the light, okay, and then produce a generator potential. So there's a lot of animals that are photoreceptors, and incredibly they are varying numbers. So here are the examples. The first one is a stigma. a euglena, okay? It has a red mass of red granules found in their body. It is swelling in the flagellum. Letter B is an ocellus, an inverted pigment cup of a flatworm. Compound I of your fly, which is containing hundreds of omnatidia. It has a convex shape, and they are precisely oriented differently for precise vision. And letter E is a complex camera-like eyes of your vertebrate eye and octopus eye. Vertebrate and octopus eye. So we also have proprioceptors, which are stretch receptors, and they are responding to mechanically induced changes by stretching, compression, bending, and tension. So they give the animal the information of movement about its body parts and their positions relatively to each other. So we have examined them in arthropods, where they are associated with appendage joints and body extensor muscles. So this is a crayfish using a stretch receptors and neurons which are attached to their muscles. So this is the muscle, and the axon of your sensory neuron send it here. Okay, it's because of some generation of your membrane potential and the inflow of sodium ions. Thermoreceptors are heat receptors, okay? Coming from the root word, it respond to temperature changes. An example of it is a horseshoe crab. So they can sense differences in environmental conditions or temperatures, okay? A horseshoe crab in this picture. How about for vertebrate sensory receptors? Yeah, we have different actions in response to stimuli, okay, in both internal and external environments. So we have chemical and physical characteristics that affect the kind of energy and molecules that carry out sensory information. So for example, the external environment consists of a medium that surrounds you, so this the external environment, and the earth that you stand and the air that you breathe. So that's how it is being affected by the nervous systems, okay, or the sensory organs. So we have lateral line system and electrical sensing for the fishes, or most of the fishes, amphibians, and platypus. So we have, they called it sensory pores in the epidermis of your skin and connecting to your canals, leading to the electroreceptors, or also called the ampullary organs. So this is the lateral line system of your jawless fish in letter A, and jawed fish and amphibians. These electroreceptors are in the epidermis and along in the head and in the body. So pores of this lateral line system will lead into canals that connect to the ampullary organ, this one that functions in electroreception and the production of the generator potentials. We have lateral line system and mechanoreceptors. They are excited with mechanical pressures or distortions such as sound, touch, or muscular contraction. So this lateral line system of cyclostomes, sharks, and more advanced fishes and amphibians includes several different kinds of hair cell mechanoreceptors. We call them neuromast, okay? These pits are along the body but not held in the body region. So these are the neuromast centers for the bony fish and the frog. Letter C is an action of a neuromast stimulation. The blue arrow will force the cap-like structure covering a group of neuromasts called the bent or distort of the shape, okay, of your of this animal. The generator will create a potential, causes an action potential in the sensory neurons. Hearing in equilibrium in air, so there are sounds, okay, when pressure waves transmit energy through some medium such as the air or water. So hearing in the air will create serious problems for mammals and for vertebrates, as middle ear transformers are sound pressure sensors. Okay, but in air, sound produces less than zero point 0.1 degrees, 0.1% of the pressure it produces in water. So we have what we call as this acoustic transformer that is being evolved into a stretched membrane. We call that either the eardrum, a tympanic membrane, or a tympanum, which is exposed to the air. So hearing is evolving in amphibians. Okay, in the ears of your frogs or neurons is consisting of a tympanum, a middle ear, and an inner ear. So they are modified and stretched into a cartilaginous ring. Take a look at this. It's a cartilaginous ring, and it vibrates with response to your movements of sounds in the on air. Okay, and touching this tympanum, called the columnella or stapes. Okay, and the membrane of the oval window, which stretches between the middle and inner ears. So this is your tympanum of the neurons. For the posterior view of your ear of the neurons, the red arrows will dictate the pathway of low frequency sounds, especially when they mate. It's really important for mating, because the females are listening to the frequency of the males' mating calls. So you have to vary it from different species and choose the right mate. So hearing and equilibrium in water. So we have semicircular canals that detect the rotational movements and other sensory patches, which help the equilibrium and balance by detecting the direction of gravitational pull. So since fishes are lacking the ear or middle ear, vibrations will pass from the water through the bones of the skull into the inner ear. Some of the few fishes have chains of bony ossicles, we call that Weberian ossicles, that pass between the swim bladder and the inner ear. So these vibrations strike the fishes and thus amplified by the swim bladder and sent through the ossicles to the skull. And these are the structures of the inner ear of your fish. The sound waves enters the mouth and transmitted to the gas in the swim bladder, causing it to expand and contract at frequencies and amplitudes corresponding to the incoming sound waves. These are the Weberian ossicles. So they have skin sensors of damaging stimuli. We call them nociceptors. Okay, for severe pain receptors. The nerve endings of your bodies of the mammals, except for in the brain and intestine. So these endings we call them nociceptors. Like severe heat and cold, irritating chemicals, strong mechanical stimuli may elicit a response from nociceptors that the brain interprets as pain or itching. We have also thermoreceptors, also bare sensory nerve endings. They are found either in epidermis or dermis, and also sensitive to either cold or warm temperatures. We call them cold or warm spots. Okay, a spot is a small area of the skin which stimulated yields a temperature sensation or of warm or cold. So for example, we have the reptiles, and they have the rattlesnakes or other pit vipers have called heat-sensitive pit organs, which is found in between the eye or the nostrils. So these are depressions that are lined with sensory epithelium containing receptor cells that respond to temperatures different from snake surroundings, and they locate the warm-blooded prey using these pit organs. Yeah, the rattlesnake is in this example, and this is the pit organ of your rattlesnake, between each eye and the nostrils. So we have skin sensors of your mechanical stimuli. So also animals are relying on tactile or touch to obtain information about the environment. So we have the sensory receptors in your skin that detect stimuli that the brain interprets, such as light touch, touch pressure, and vibration. Also, mammals have a echolocation. Okay, so they're using this distance and depth of the sound waves using this sonar biosonar. They emit high frequency sounds like for the bats, and then determine how long it will take for the sound to return after bouncing off some objects in the environment. So they will be using this to locate using the sound waves. So we have locomotion for non-muscular and muscular systems. For smell, okay, olfaction, using the olfactory neurons to the roof of the vertebrate nasal cavity. These cells, which are a specialized ending of the fibers that make up the olfactory nerve. So for first cranial nerve, it ally most among the supporting epithelial cells. They are densely packed, and for the dog, they have 40 million olfactory receptor cells per square centimeter. Okay, so these are the olfactory nerves and the olfactory neurons receptor cells in their human nasal passageway. For smell, olfactory senses are also better developed in reptiles than in amphibians. So there, in addition to having more olfactory epithelium, most reptiles except crocodiles have blind ending pouches. We call them Jacobson or vomeronasal organs. So they are developed in snakes and lizards. These protrusible forked tongues of snake and lizards are accessory organs for airborne chemicals. So they have to test the chemicals or the air if there is a toxic chemicals that are coming into their way. For the smell, this one is a Jacobson vomeronasal organ. Oral cavity and nasal cavity, lacrimal ducts in the eye of your reptile. For taste, so you have receptors for taste. We call them gustation or chemoreceptors. They may be on the surface of your animal. Okay, so we have in the mammals, mammalian tongue, we have this papilla, giving the bumpy appearance. In this example, papilla, taste buds and sensory neurons and connective tissues. For vision, a primary sense that vertebrates in a light-filled environment use consequently. So we have capable of interpreting visual image, and for the next figure, it has an eyeball, a lens, a sclera, a tough outer coat, a choroid layer, a pigmental layer, and an inner retina containing many light-sensitive receptors. We call them photoreceptors, as we discussed from the invertebrates earlier. And this is the anatomy of your human eyeball. Light passes through the transparent cornea here, and the lens focuses the light into the rear surface of your eye, the retina, this one, and at the fovea centralis. And the retina is rich in pigments and rods and cones. Also with the median eye of your reptiles, okay, it has a connection to the lateral eyes. Next we have magnetoreceptions. Okay, the homing salmon is an example of magnetoreception, has a magnetoreception ability. They can detect magnetic fields. Okay, so these are extensively studied. Okay, these are not yet understood among animals and remains most elusive for its sensory modalities. And this is a paper in homing Pacific salmon, the mechanisms and ecological basis, which is studying on the magnetoreceptors. So thank you so much for listening, and if you have any more questions, don't forget to drop and comment down, subscribe this channel, and stay safe. Thank you so much.